Protist Diversity. Section 13.3. By the end of this video, you will be able to describe important pathogenic species of protists and describe the roles of protists as food sources and as decomposers. With the advent of DNA sequencing, the relationships among protist groups and between protist groups and other eukaryotes are beginning to become clearer. Many relationships that were based on morphological similarities are being replaced by new relationships based on genetic similarities. Protists that exhibit similar morphological features may have evolved analogous structures because of similar selective pressures, rather than because of recent common ancestry. This phenomenon is called convergent evolution. It is one reason why protist classification is so challenging. The emerging classification scheme groups the entire domain Eukaryota into 6 supergroups that contain all of the protists as well as animals, plants, and fungi. These include the excavata, chromalveolata, rhizaria, archaeplastida, amoebozoa, and opistheconta. The supergroups are believed to be monophyletic. All organisms within each supergroup are believed to have evolved from a single common ancestor, and thus all members are most closely related to each other than to organisms outside that group. There is still evidence lacking for the monophyly of some groups. Figure 13.15. Protists appear in all 6 eukaryotic supergroups. Welcome back to Concepts of Biology. We'll continue with human pathogens. Many protists are pathogenic parasites that must infect other organisms to survive and propagate. Protist parasites include the causative agents of malaria, African sleeping sickness, and waterborne gastroenteritis in humans. Other protist pathogens prey on plants, effecting mass destruction of food crops. Plasmodium species. Members of the genus Plasmodium must infect a mosquito and a vertebrate to complete their life cycle. In vertebrates, the parasite develops in liver cells and goes on to infect red blood cells, bursting from and destroying the blood cells with each asexual replication cycle. Of the 4 Plasmodium species known to infect humans, Plasmodium falciparum accounts for 50% of all malaria cases and is the primary cause of disease-related fatalities in tropical regions of the world. In 2010, it was estimated that malaria caused between 0.5 and 1 million deaths, mostly in African children. During the course of malaria, Plasmodium falciparum can infect and destroy more than 1/2 of a human's circulating blood cells, leading to severe anemia. In response to waste products released as the parasites burst from infected blood cells, the host immune system mounts a massive inflammatory response with delirium-inducing fever episodes as parasites destroy red blood cells, spilling parasite waste into the bloodstream. Plasmodium falciparum is transmitted to humans by the African malaria mosquito, Anopheles gambiae. Techniques to kill, sterilize, or avoid exposure to this highly aggressive mosquito species are crucial to malaria control. Figure 13.16. This light micrograph shows a 100-time magnification of red blood cells infected with Plasmodium falciparum, seen as purple. Trypanosomes. Trypanosome brucei, now being called T. brucei, the parasite that is responsible for African sleeping sickness, confounds the human immune system by changing its thick layer of surface glycoproteins with each infectious cycle. The glycoproteins are identified by the immune system as foreign matter, and a specific antibody defense is mounted against the parasite. However, T. brucei has thousands of possible antigens, and with each subsequent generation, the protist switches to a glycoprotein coating with a different molecular structure. In this way, T. brucei is capable of replicating continuously without the immune system ever succeeding in clearing the parasite. Without treatment, African sleeping sickness leads invariably to death because of damage it does to the nervous system. During epidemic periods, mortality from the disease can be high. Greater surveillance and control measures have led to a reduction in reported cases, some of the lowest numbers reported in 50 years. Fewer than 10,000 cases in all of sub-Saharan Africa have happened since 2009. In Latin America, another species in the genus Trypanosome cruzi is responsible for Chagas disease. T. cruzi infections are mainly caused by a blood-sucking bug. The parasite inhabits heart and digestive system tissues in the chronic phase of infection, leading to malnutrition and heart failure caused by abnormal heart rhythms. An estimated 10 million people are infected with Chagas disease, which caused 10,000 deaths in 2008. Figure 13.17. Trypanosomes are shown in this light micrograph among red blood cells. Plant parasites. Protist parasites of terrestrial plants include agents that destroy food crops. The oomycete Plasmopara viticola parasitizes grape plants, causing a disease called downy mildew. Grape plants infected with P. viticola appear stunted and have discolored, withered leaves. The spread of downy mildew caused the near collapse of the French wine industry in the 19th century. Figure 13.18. The downy and powdery mildews of this grape leaf are caused by an infection of Plasmopara viticola. This potato exhibits the results of an infection with Plasmopara infestans, the potato late blight. Phytophthora infestans is an oomycete responsible for potato late blight, which causes potato stalks and stems to decay into black slime. Widespread potato blight caused by Phytophthora infestans precipitated the well-known Irish potato famine in the 19th century that claimed the lives of approximately 1 million people and led to the emigration from Ireland of at least 1 million more. Late blight continues to plague potato crops in certain parts of the United States and Russia, wiping out as much as 70% of crops when no pesticides are applied. Share the link. We want these free textbook resources to be available to other students. All right, let's get back to reading. Beneficial protists. Protists play critically important ecological roles as producers, particularly in the world's oceans. They are equally important on the other end of food webs as decomposers. Protists as food sources. Protists are essential sources of nutrition for many other organisms. In some cases, as in plankton, protists are consumed directly. Alternatively, photosynthetic protists serve as producers of nutrition for other organisms by carbon fixation. For instance, photosynthetic dinoflagellates called zooxanthellae pass on most of their energy to the coral polyps that house them. In this mutually beneficial relationship, the polyps provide a protective environment and nutrients for the zooxanthellae. The polyps secrete the calcium carbonate that builds coral reefs. Without dinoflagellate symbionts, corals lose algae pigments in a process called coral bleaching, and they eventually die. This explains why reef-building corals do not reside in waters deeper than 20 meters. Not enough light reaches those depths for dinoflagellates to photosynthesize. Figure 13.19. Coral polyps obtain nutrition through a symbiotic relationship with dinoflagellates. Protists themselves and their products of photosynthesis are essential, directly or indirectly, to the survival of organisms ranging from bacteria to mammals. As primary producers, protists feed a large proportion of the world's aquatic species. On land, terrestrial plants serve as primary producers. In fact, approximately 1/4 of the world's photosynthesis is conducted by protists, particularly dinoflagellates, diatoms, and multicellular algae. Protists do not create food sources only for sea-dwelling organisms. For instance, certain anaerobic species exist in the digestive tracts of termites and wood-eating cockroaches, where they contribute to digesting cellulose ingested by these insects as they bore through wood. The actual enzyme used to digest the cellulose is actually produced by bacteria living within the protist cells. The termite provides the food source to the protist and its bacteria, and the protist and bacteria provide nutrients to the termite by breaking down the cellulose. Agents of decomposition. Many fungus-like protists are saprobes, organisms that feed on dead organisms or the waste matter produced by organisms. Saprophyte is an equivalent term, and they are specialized to absorb nutrients from non-living organic matter. For instance, many types of oomycetes grow on dead animals or algae. Saprobic protists have the essential function of returning inorganic nutrients to the soil and water. This process allows for new plant growth, which in turn generates sustenance for other organisms along the food chain. Indeed, without saprobic species such as protists, fungi, and bacteria, life would cease to exist as all organic carbon become tied up in dead organisms. Now on to Section 13.4, Fungi.